Okay, can you all hear me? Finally got my internet back.
I can hear you.
Great, thank you. I'm going to try to get back into where we were and keep going. The other slide, there we go. So I think I was talking about the glycocalyc structures. Let me get back out of this and there we go. So we're definitely having some interesting Zoom connections this week. I can say that. So. Okay, now, so I was telling about the glycocalyc structures. The our immune system is typically, going to take about 10 days to 2 weeks to respond to a brand new infection. If you get the flu, for example, you're going to feel symptomatic for about 10 days. You're going to feel really bad, maybe from the third or fourth day on to about the 8th day, then you're going to start feeling better because it takes your immune system that long to respond to the flu virus. Once it responds, though, it has a massive response and destroys the virus. Cancer cells take advantage of this. Cancer cells are going to change their glycocalyc structures rapidly enough so that every time your immune system responds to their glycocalyx, they've changed it and they have to start over, so they can stay ahead of the immune response simply by constantly modifying their own glycocalyx. So moving on, let's talk about cell junctions. Now, something else that proteins do is help hold our cells together. Now, cells are bound together to each other 3 different ways. We have what are known as tight junctions, where nothing can get through the cell membrane. We have desmosomes, where we have a great strength but great flexibility. And we have gap junctions, where we literally have openings between the membrane from one cell to another. So the tight junctions show up in our stomach to keep the contents of the stomach from leaking out into the surrounding tissues, because there's stomach acid there. It's hydrochloric acid in our stomach. And so we don't want that to get out of the stomach. So we have the cells of the stomach held together with tight junctions. The membranes essentially are stitched together. So it's almost like they're sewn together. And so that it is an impenetrable lining in here so that nothing can get through. The stomach acids, the contents of the stomach, it can't leak out. So we use this particular very powerful connection here. Now, the second type are the desmosomes. The desmosomes look like bolts or rivets that go across the membrane from one cell to another. They're anchored in place on both sides. They're very, very powerful connections, but they're also very flexible. And if you look at them under the microscope, it would look like almost like a zipper in here. The bolts are keratin, which are a waterproof protein, and they're very strong, but they're also flexible. So imagine very powerful bolts that also have some give. We find this where we want to have great strength, but we also want to have flexibility. In our heart, for example, our heart has to contract every 0.7 of a second, and Every cell in our heart has to contract at exactly the same time with exactly the same force. So we have to have, the heart contracts vigorously. The cells all have to hold together as we have this vigorous contraction. And then we relax, and then we do it again 7/10 of a second later, and we do it again in another 7/10 of a second. And we do this for 70, 80, 90, 100 years or more in individuals. So we are going to see this happening again and again and again. So we have to have great strength, but we also have to have great flexibility. We can't restrict the force of the contraction. The force of the contraction has to be exactly the same in every cell. And so we have this give in here. We would have this same type of connections in our skin cells, so that we have give and flexibility. There's great strength, but also great flexibility. The third category are the gap junctions. The gap junctions are openings between the cell membranes of adjacent cells. We use this for the movement of ions to go from cell to cell. We also use it for nerve impulses. It's much easier for a cell to send a nerve impulse through these gap junctions than it is to run across the top of the cell and go down inside the cell and then back up to the other side. It's much faster. This is, the cardiac muscle cells use gap junctions. So we have rapid movement of a nerve impulse across the heart, so that every cell is stimulated at exactly the same time to contract together at exactly the same time with exactly the same amount of force. So when the heart contracts, it acts as if it's one great big giant cell. There are no, it's not like in our arms and legs and our skeletal muscle where we control the strength of the contraction by how many cells in the muscle are going to get activated. Here in the heart, everything gets stimulated and everything contracts together. So let's shift gears and take a look at the membrane itself. Important point here, we've discussed this in lab, and I'll give you some greater insight on this. Cells love things that don't cost them any energy. Cells view energy as its currency, as its cost. And anything that's free, like most of us, if it's free, we all like free stuff. So cells do too. They like free movement across their membrane. They don't want to have to work to get stuff into the cell. So they call it passive transport. It still moves into the cell or out of the cell, but the cell doesn't have to pay a price, and the price is always energy. So with passive transport, there is no price. Now, passive transport is either diffusion or filtration. We'll get to filtration here in a second. Diffusion is simple diffusion, movement from a high concentration to a low concentration. Very simply that. We have facilitated diffusion where we have to use different types of proteins. And we come to the proteins again, but different types of proteins to assist the movement, the free movement across a membrane. And then, of course, there's osmosis, the movement of water across a membrane. Filtration is where you drive, where the capillary actually allows certain materials to leave the plasma, and be captured somewhere else. The capillaries can serve as a filter or a sieve. Our kidneys depend on filter out our blood, and they pass plasma through tiny openings in the walls of the capillary. And with whatever can pass through those openings, that's small enough to pass through those openings, when the plasma goes through, will go along with it. Things like sodium and potassium and maybe the occasional white blood cell, but little tiny things like amino acids. Um, Glucose shouldn't be going through that membrane, shouldn't be filtered, but sometimes it is. So, okay. So simple diffusion is a free ride. It lets things pass across the membrane of the cell without any effort of the cell. It can go into the cell or out of the cell. Diffusion, so we're seeing an example here. Diffusion is always the movement of molecules from a high concentration to a low concentration. See, everything has random motion. So if you drop a dye pellet into water, the water molecules are moving around randomly. And so the dye pellet, the dye molecules will start moving around randomly too, and they will gradually move from a region of high concentration to a low concentration until they reach equilibrium. So they don't know it's equilibrium, they're just all moving around. And then, but the material is spread evenly in our beaker here. We can see we go from the we always go from the high concentration to the low concentration. Diffusion outside the body is driven by the particle size and by temperature. Large molecules diffuse slower than small molecules. Diffusion occurs at a faster rate when it's warmer, when the temperature is up, because the water molecules are moving around faster. So, if you consider the tea and sugar analogy, sugar goes into solution in iced tea when the tea is hot at a faster rate than if the tea is cold, because the water molecules are moving faster. The speed of diffusion is driven by the size of the molecule and by temperature. You wouldn't throw a 5-pound block of sugar into a container of tea and expect it to dissolve quickly. You use the granulated sugar. So, for a cell to function by diffusion, the cell doesn't require any of its own energy. It moves simply from a high concentration to a low concentration. But the cell membrane doesn't like diffusion because not everything, it likes diffusion. It just doesn't like some of the things trying to diffuse across the membrane. The plasma membrane is selectively permeable. Things that are water soluble don't get across. Molecules that are charged don't get across. Large molecules don't get across, at least not on their own. So simple diffusion from a high concentration to a low concentration doesn't work in our cells for a lot of things. Things that are not charged can slide across. Oxygen can go across, carbon dioxide can go across, they're very tiny. The fat-soluble vitamins go across very easily. Cholesterol goes across very easily. Anything that doesn't have a charge, non-polar, that leaves out water, because water is polar, leaves out the ions, because the ions have charges, positive and negative charges. The ions don't go through the membrane. And so we're restricting. But we still need those particular substances. We have to have ways to get those substances in. Simple diffusion is not going to get sodium into the cell. Simple diffusion is not going to get glucose into the cell or water. We have to have assistance. We have to have a way to facilitate their movement. The problem with the cell membrane, you have a burn patient with third-degree burns. If you've ever seen a third-degree burn, you know what I'm talking about when I say that it looks an awful lot like barbecue, because the skin is charred and burnt and essentially cooked, if you will. We're talking burned down to the bone. It's a horrific sight to see tissue look like that. The nearest analogy is to say, like if you got something that you grilled or something that you put on the smoker, that's about the best I can come up with as far as what human tissue would look like. When you have that much damage during a third-degree burn, burn down to the bone, you have destroyed the cell membranes of many cells, and you've lost the protection of those cell membranes. And so a patient with severe burns is going to be constantly oozing water and nutrients and ions out of their cells, out to surrounding tissue or leaking out of the body. And so you're, among other things, you're busy pumping fluids back in to make up for the fluid loss. We can't afford to lose that kind of fluid. It also creates that movement of fluid also creates an ideal location for bacterial infection to occur, because the damaged cells are warm and dark and moist, and lots of nutrients are flooding out of there. Bacteria love that kind of stuff. So it's a race to try to keep the fluids coming in as they're leaking out. The only advantage that a burn patient has with burns to that extent is that they usually don't hurt, because the pain receptors have all been burned away. But not all of them. But there is usually less pain than you would imagine, simply because of damage to the pain receptors in the skin. Facilitated diffusion is how the cell gets around the issue of these large molecules or these charged molecules or these water-soluble molecules. With facilitated diffusion, you have some sort of specialized channel or carrier designed to move things across the membrane. If it's a channel protein, it's often just a tube. Like you see up here, this is a channel protein. It's a tube through the membrane. Water frequently uses what's known as an aquaporin, a water pore, if you will, to let water go into the cell or water leaves the cell. So it's how we can get around the hydrophobic nature of the cell membrane. So it's a pretty simple tube. Now, other proteins are the carrier proteins. And the carrier proteins are pretty specific. This example here is a carrier protein. The protein is designed to accept one molecule type only. Maybe this is sodium. Maybe it's potassium. But only sodium or potassium or whatever molecule it is will fit right there on the structure of the carrier protein. And that when it lands there, the carrier protein will change its shape. Doesn't cost anything energy-wise. It will change its shape and direct the molecule through the protein out to the other side. These carrier proteins are usually pretty specific. Sodium carrier proteins only work for sodium, not potassium. Potassium has to have its own. Glucose uses a carrier protein, the sugar, and it works for sugar, but not for any of the ions. What works for glucose won't work for water. So these carrier proteins are very specific. They keep the membrane closed when they need to, and only let these specific molecules through when they encounter the appropriate protein. They are specific. They can let sugars come through. These carrier proteins let sugars come through. They can let amino acids come through. They can allow certain types of hormones that come through that are not fat soluble, but are not so big that they couldn't possibly fit. Glucose will go through here, but nothing else will. The carrier proteins will simply change their shape to allow the molecule to pass through. That's how we, among other things, that's how we get sugar into our cells, thanks to insulin. Insulin tells the cell to make carrier proteins just for glucose. Glucose can go in the cell. And then once we've established homeostasis, once the glucose levels in the blood, once the blood sugar levels come back to normal, we stop making insulin and we stop making carrier proteins. We don't leave them there. We just don't mix it. They disintegrate, and then we make new ones. Now, the channel proteins, let me make a point about the channel proteins too. Channel proteins do a great job with water. They can also do a good job with different ions. Sodium and potassium can also take advantage of the channel proteins. Now, some of these channel proteins are always going to be open. We call them leakage channels. Aquaporins are usually always open. Water goes through, water comes out, water goes in, water goes out. That's fine. Sodium can come in, potassium can go out, potassium can come in, sodium can go out. So they're going to be very specific here for the movement of these ions too. Now, if it's a leakage channel, it's always going to be open. Now, other channels can have what we call a gate on there, and the gate is going to stay shut. The gate is going to be controlled by a molecule, some sort of chemical molecule that will open the gate or close the gate. We use it as a signal to open or close the gate. We also can open or close the gate electrically. Our bodies are electrically charged, and we can control the gate by sending a charge to the gate and say, open up or shut down. Those are pretty specific. We're not going to talk about them right now. We will later on, because this is how we tell muscles when to contract, by using these electrically charged gates in our bodies. So, okay, now. So to summarize, the plasma membrane is selectively permeable. Some things go through, some things don't. Movement across the membrane is either passive or it's active. And we talked about the passive processes: diffusion, facilitated diffusion. I'll mention osmosis here in a second. The active processes always require the use of ATP, always require energy. The whole thing about an active process is that you have to move, it doesn't matter what you're doing, but you're moving against a concentration gradient. You're essentially pumping uphill, if you will, because with active transport, while normally the substance leaks into a cell passively, you have to pump something back out of the cell against the concentration gradient. And that's active transport. And that requires energy in the form of ATP. So osmosis, passive, doesn't require energy. Now, instead of ions, we are moving water across the membrane. Now, water is being driven by osmotic pressure. The osmotic pressure is caused by the solute concentrations. It's the presence of the solutes in the water that are going to drive the movement of water across the membrane. Solute concentration. Water goes across the membrane, driven by its osmotic pressure. Term here is osmolarity. This is an important point to remember. Osmolarity is a measure of the total number of solute particles in a solution. If you make up a 10% salt solution, the osmolarity of that solution is based on how many salt particles, how many sodium and chloride particles are in that solution. So 10% salt has X number of solute particles. 20% salt has X number more solute particles. The concentration is every solution is going to be different. The concentration is going to be variable. The number of solute particles are going to be different. But it's the number of solute particles present that drives the movement of water. If you have a solution that is let me just say what it says here. If the solute concentration goes up, if it increases, the water concentration goes down. A 10% sodium chloride solution is 90% water. But if you increase the solute concentration, go from 10% to 40%, now your water level has dropped. If a 10% sodium chloride solution is 90% water, but a 40% sodium chloride solution is only 60% water. Now, if you have a membrane and you have two different solutions, two different concentrations of a solution, one on either side of the membrane, water will go across the membrane from the side with the greatest water concentration. So if you have one side of the membrane has a 10% salt, which is 90% water, and the other side is a 40% salt, which is only 60% water, the 90% water side is the higher water side. Water always goes from the higher water to the lower water. So a 10% salt solution is 90% water, and a 40% salt solution is only 60% water. Water goes from the 90% to the 60%. It will go across the membrane. So water goes from the areas of high water to low water. And high water equals low solute. If you're high, if you're 10% salt and 90% water, that's the high water is that's a high water concentration. Especially if you're comparing it to 60% water and 40% salt. So water will go across from the high water side, it will go across a membrane to the low water side until we reach equilibrium. Until the solute concentrations are exactly the same on both sides. And that makes the water concentrations exactly the same on both sides. So water will continue to move across the membrane from the high water side to the low water side until we reach equilibrium. Once we reach equilibrium, then there is no net movement of water anymore. And that's what happens in our cells. Our cells are usually at equilibrium because the salt concentration and the salt concentration of the fluid outside the cells is usually exactly the same as the salt concentration of the fluid inside the cells. The interstitial fluid, the fluid outside the cells, is 0.9% sodium chloride. The intracellular fluid, the fluid in the cytoplasm, is also 0.9% sodium chloride. So there's really no change. There's zero net movement of water across the membrane. The only time we would see water moving across the membrane in large quantities is if we become either overhydrated or dehydrated. Because if we're overhydrated, we have too much water outside the cells, and that water will enter the cells, high water to low water. If we're dehydrated, we've lost water in the interstitial fluid, there's more water in the cells will send that water out. But that usually doesn't happen because we're usually not dehydrated and we're usually not overhydrated. It can happen, but it's usually we maintain a net balance where there's no water going back and forth. Now, water moves. When it does move, we have water inside the cell pushing out, and we have water outside the cell pushing in, but there is zero net pressure. So hydrostatic pressure inside the cell is pushing out, and it's balanced by the osmotic pressure pushing in. The only thing that drives movement in or out of the cell is when we have more solutes. The greater the solute concentration, the greater the pressure we're going to have to move. Okay.
The plasma membrane is permeable to water molecules, and the more much water into the
Okay, we're going to stop here, and there'll be two videos posted today, because we had, before the power loss and after the power loss. So if anybody has any questions on anything, shoot me an email, and I will get back to you later on today. So I'm going to get us out of here because I've got the next class getting ready to start. So I will see you later on lab today. We are going to look at the appendicular skeleton. I think that's where we are. So, okay, I'll see you all later.